Alright guys, welcome back to Underground Science, and in this video we'll try to cover the steps of an action potential. Alright, hopefully it doesn't get too long, or it's fine, we'll just continue in the next video. Alright, so let's go and get started. So remember from our neuron playlist and the nervous system playlist, right, all the videos. So let's just go ahead and just refresh our memory with the basic structures of the dendrite. Or basic structures of the neuron. Sorry, I was writing dendrite here. So these are the dendrites, all right? And we have the cell body here, or the soma, right? So the cell body slash soma. And again, inside our cell body, we do have a nucleus, right? We do have a nucleus with its DNA, and we do have a mitochondria and other organelles too. But just to save time, let's just go and draw the mitochondria because it's amazing, right? The powerhouse of the cell. And then we have our axon, right? So we have our axon in orange. So this whole area right here in orange that you see hidden behind the myelin is the axon. And this is the myelin sheath that we might get to in this video about why it's happening, why the myelin sheath helps conduct action potentials faster, right? Our myelin sheath mostly made up of fats and a little protein. Then we have our axon terminal. All right, we have our axon terminal. And here we have our synaptic cleft. All right, so here we have our synaptic cleft. So the open space between our presynaptic neuron and our postsynaptic cell is our synaptic cleft. All right, this is where the neurotransmitters are released. All right, so that's the basic structures, and so let's go and get started. So let's talk about what happens during an action potential. So first, let's start to label our graph here, right? So right here is going to be about our resting membrane potential. Generally, for our neurons, right, our resting membrane potential is going to be around negative 70 millivolts. All right, and then what's going to happen is that if the graded potentials and the temporal and spatial summation is great enough, right, from the graded potentials, then our total electricity, or in voltage in millivolts near the axon hillock, can reach the threshold potential. So let's go and draw that out. So the threshold potential, let's say, is about right here. Let's say our threshold potential, it's going to be about between negative 45 millivolts to about negative 50 millivolts, all right? So in some sources you'll see negative 45, some will say negative 50, so let's just say it's around there. All right, so that's our threshold potential. And then once it hits our threshold potential, guys, it's an all or none phenomenon. The action potential will rise up all the way to about positive 35 millivolts. All right, so let me go and erase this out for you guys since we already went over it. So it's gonna rise up to about positive 35 millivolts, right? That's it. So it's an all or none phenomenon. So about positive 35 millivolts. And so let's go and label these parts. So this, remember, negative 70 about is our resting membrane potential. Resting membrane, excuse me, potential. All right. And then negative 45 millivolts to about negative 50 is where we see our threshold potential, right? Our threshold potential. All right. And then at about positive 35 is our limit, where we go at the maximum limit of depolarization. Then we repolarize, and then we hyperpolarize into a refractory period, and then we start again. All right, so let's talk about this. So remember in our graded potentials video, we said that if we have excitatory stimulus, right, many stimuli, it's going to add up as temporal and spatial summation. Okay, so let's say we have a lot of positive stimuli coming here, right, on our dendrites. We're picking up on our receptors of our dendrites, and it's trying to bring it in. Well, these positive stimuli, right, will cause temporal and spatial summation, all right? So temporal and spatial summation. And as these electric graded potentials reach the axon hillock, Oh crap, we forgot to mention it. The axon hillock starts right before the axon, right, where the action potential is generated, or the trigger zone. So the axon hillock is also called the trigger zone. All right, so let's go and write that down. Axon hillock. So as the graded potentials reach the axon hillock, the graded potentials, unlike the action potential, right, are sometimes, so the graded potentials generally will actually decrease in magnitude and size, right, as it reaches the axon hillock. So it just because some excitatory stimulus comes here doesn't mean our threshold potential is gonna be reached, and doesn't mean we're gonna fire an action potential. It has to be many positive stimuli, many excitatory stimuli, in order to reach the action potential at a certain threshold potential, right, at about negative 45 to 50 millivolts. Because remember, if something's very excitatory here, by the time it gets to the axon hillock, it's not very big in magnitude, right? So it's not going to fire the action potential if it doesn't reach negative 45 to negative 50 millivolts. So we need a lot of positive stimuli so we can get to the threshold potential, all right? So what happens here is that as the positive stimuli build up, right? So if we zoom in to one of these, the tip of these dendrites, we're actually going to have the dendrite coming here, and then we're going to have a patient receptor here on the dendrite that's actually going to receive those neurotransmitter signals from the whatever. So if we counted this as a postsynaptic neuron, then if there's a presynaptic neuron here and they let go of their neurotransmitters, if it's excitatory, right, we're going to receive those positive signals here, so the neurotransmitters are going to bind. Then what's going to happen here is that we're going to sort of have a channel here where we'll see mostly sodium cations that will flow through this channel, right? So we'll see sodium cations that will flow through this channel and actually go through the soma and to the axon hillock. And these sodium cations that will flow will actually, because we have a positive charge on sodium, these positive charges can add up, right? Temporal summation, and it can add up. And by the time it reaches the axon hillock, if our threshold potential is reached, right? If our threshold potential is reached, then the axon hillock or the trigger zone will fire an action potential, right? And there'll be many action potentials fired throughout the axon. So let's go and write that down. So right here, we're at negative 70, right? So in the beginning, we might get some, like maybe some excitatory signals, maybe a few sodiums flowing into the neuron, right? But it's not much. And then we have many more excitatory signals, so we add up that in terms of temporal and spatial summation, right? And more sodium channels, sodium ions start flowing in, and then bam, right here we hit it, right? We just hit the threshold potential, and now we're gonna have that all-in-one phenomenon where we're gonna fire that action potential. So these temporal summations right here and spatial summations that involve the neuron getting slowly depolarized, just slightly, right? Involve the sodium. It was because of the sodium cations that were coming in. So that means that right here is because of these sodium cations. So let me go and label that out. So when we go from negative 70 to threshold potential, right, in the beginning, before we actually hit that threshold potential, that's because of the graded potentials that our neuron receives, all right? So that's because of the graded potentials that our neuron receives, right? And mostly because of the sodium cations that flow into the neuron as the positive excitatory neurotransmitters bind to the receptors of the dendrites. All right, now, so let's go and draw that. Let's go and draw this. So we hit that threshold potential. So let's go and draw it less steep, like we're slowly going up, right? So let's represent the temporal spatial summation that we just did right here. So we're going right, temporal spatial summation, and bam. Right now we're going to fire an action potential all the way to positive 35 millivolts. First things first, let's go and note down that this part that we just fired up, right? When our neuron cell gets more positive, we call that the depolarization state. So our neuron's getting more depolarized. So this is our depolarization state of our action potential. All right, sorry about that random mark. All right, so that's our depolarization state. So again, these are the graded potentials. Bam, we hit the threshold potential. We fire that those action potential, right? Our trigger zone, our action on hillock sets those action potentials off. Now, how does it get way more positive right here like that? Well, let's, you know what, let's just go and pick this up in the next video. All right, so see you guys there.